Hello everyone. So today we're going to start with our next session of life processes, the sixth session. This is a topic of 10th class, which is excretory system. So excretory system, if we deal of, is a system which involves the release of the nitrogenous waste from the body. These nitrogenous waste, they may be the ammonia, they may be the urea, they may be the uric acid, creatinine. These are the various nitrogenous wastes that are formed in our body due to the metabolic reactions. Now, our body needs to expel them out. For expelling these things, the excretory system has special structures, and these special structures, they are packed together in an organ. So let's deal with what kind of organ this excretory system has. What are the different components of that excretory system? So dealing up with the first one, that is the excretory system comprises of the kidneys. The kidneys, these are a pair of structure, the right kidney and the left kidney. And as I told you, the right part of your hand is the left part of your diagram. Similarly, the left part of your hand is the right part of your kidney. So if I see, this is my right kidney. Now this right kidney and this left kidney, if I place my hand, you can see they are comparatively up and down. Similar is the case of this particular kidney. You can see here that the right kidney is slightly lower than the left kidney. The kidneys, they are bean-shaped structure. Now these kidneys, they're richly supplied with the vessels. And these blood vessels are the arteries and the veins. Because artery is the one which is carrying the oxygenated blood. Artery is the one which is carrying all the food material together. So that food material is traveling through the arteries, but that artery is also having some of the waste material which was being formed in the liver. So liver now needs to expel out that particular dirty thing, the waste thing from the body, or either it will make our body, suffering from various diseases. So in that case, that artery that is here, as stated, that arteries, they are red in color when shown in the diagram. So this is the artery, the aorta, while the blue color is the vena cava. Now the arteries carrying the blood enters into this kidney-shaped structures. And after that, you can see this yellow-shaped structures coming down. Now, kidney, they make the urine. Now this urine, so form is collected and drained down into a tubular structure. Now this tubular structure is known as ureter. Now this ureter, these are tube-like structures that join together into a muscular bag. Now this muscular bag is known as urinary bladder. Bladder is a sac, muscular sac, which is in control with our brain. It is the urge of urination that can be controlled by our body. So that means the nervous system is actually controlling this bladder. The next is the urethra, by which the urine is expelled out. Now the length of the urethra, it varies in case of the males and females. For males, it is larger in size, and for females, it is smaller in size. The function of the urethra in males is double. It carries up the urine and is also an organ which is responsible for the transfer of the sperm. So it is connected to the urinogenital, that means reproductive system. Now dealing with this particular structure, that the urinogenital system, we are just dealing with the excretory system. So this excretory system, the kidneys, as you can see here, in these kidneys, there are the triangular shaped structures, and these triangular shaped structures, they are known as the medulla. If I consider as this a kidney, now you can see the space between this hand and the triangle is 3D space over here. This space is nothing but cortex, while this triangular structure is known as medulla. So cortex and medulla. Now this cortex and medulla, they drain up. They have a special structure known as nephrons. So unit of the excretory system is actually a nephron, which is going to carry out the work like we had in the lungs. Though the air was being carried out by the respiratory system, the nose, the nasal cavity, it goes into the bronchi and bronchioles, and finally is into a sac-like structure known as alveoli. Similar is the case of this particular kidney. When I talk of this kidney is having the nephrons. Have you imagined when you open up the packet of noodles, the Maggi noodles? Same is the case of the kidney. The kidney is packed, the nephrons, they are packed into the kidney like noodles, they are packed in the packet of the Maggi noodles. So every noodle here can be considered as a nephron. So when we deal with this nephron, the nephron you can see is a coiled structure. This coiled structure is having different components. The first component, if I talk of, is a blood vessel, which is draining its blood into the nephron. Now, this blood vessel is nothing but an artery, and this artery is in the form of arteriole. The larger aorta breaks down into the arteries they break down into the arteriole, and they supply this oxygenated blood along with the food material to the different organs. So here you can see in the Bowman's capsule, this particular afferent arteriole is draining its content. Now this afferent arteriole, it breaks down into the small capillaries. Now these capillaries, the tuft or the bunch, TUFT, tuft or bunch of the capillaries is termed as in glomerulus. Now, when I'm talking of this glomerulus, I can see that this glomerulus further gets combined and results in the formation of another tube, and that's an efferent arteriole. So efferent arteriole is the one which is bringing the blood into the glomerulus, into this particular structure, and finally is leading to a structure which is finally leading to a tube known as efferent arteriole. So the blood entering here moves into the glomerulus. Now this glomerulus, if I talk of, glomerulus is simply a sieve. Just imagine you are making a tea. You are adding n number of things in that. You had added water, you had added milk, you had added sugar, ginger, you had added some another spices to it. You have added tea leaves to it. Now, when you drink tea, do you drink whole amount of that? No, obviously no. What you do? You try to sieve all the contents into a cup-like structure, into a cup, into a glass, whatever the thing is. So when you are sieving it, you get the essence of everything. But the large size particles like the tea leaves or the other things, they are left behind. Similar is the case of this afferent arteriole. The afferent arteriole, when it takes the blood into the glomerulus, when it takes the blood into the glomerulus, it acts as a sieve. It helps in the filtration of this particular structures. Now this filtration of the structure makes the content to move into this cup-like structure known as Bowman's capsule. Now, this Bowman's capsule is, and the glomerulus, they are made up of special cells known as epithelial cells. Now these epithelial cells, they are very much efficient in the filtration process. Now this Bowman's capsule, it leads to a coil structure. As I said, the Maggi noodles, the noodles. So this is the noodle-like structure. You can see the curve, the wave-like structure. So this wave-like structure is known as convoluted tubule. You can see the word convoluted here. So this is my Bowman capsule. If I say this is the convoluted tubule, this convoluted tubule now moves like this. You can see the structure is somewhere somewhat depicted like this. Now this is also a convoluted tubule, this is also a convoluted tubule. Now these two convoluted tubule, ones which is near to this Bowman capsule, is known as the proximal. पास में है तो proximal. Now this is comparatively far off from this. अगर आप यहाँ पे देखें, so this is comparatively far off. यहाँ से दूर है. You can see here it is far off. So this structure is known as distal convoluted tubule. So P for pass, P for proximal, D से दूर, D से distal. Now this distal convoluted tubule now leads to a U-shaped structure. Now this U-shaped structure, if I'm saying, is known as loop of Henle. Since Henle was a scientist who described it, so it's known as loop of Henle. Now, when I talk of this loop of Henle, the Henle, then the one you can see is going down. Now, as it is going down, we give it a name as descending limb of loop of Henle. Limb means hand. So descending means downwards. So this is descending limb of loop of Henle. Then comes a U-shaped structure which is now going up. Now this movement of the structure up is known as the ascending limb of loop of Henle, which joins together to the distal convoluted tubule. Now this distal convoluted tubule is joining a long duct, and this is the collecting duct. Now this collecting duct, if I'm talking of, is actually getting the content from various nephrons. As if I say this is the collecting duct, this may be one of the nephron, one of the DCT, this may be the other DCT, this may be the other DCT. So there are n number of DCTs connected to this particular collecting duct. Now various collecting duct, you can see they can drain all their content into this particular ureter. So this is how the urine is collected. So this is the structure of a nephron. Now you can see the efferent arteriole, the second one, the efferent arteriole, it is actually giving this content. You can see this, the blood is actually, this efferent arteriole, it breaks into capillaries and surrounds whole of this particular nephron. Now this surrounding of the whole nephron by these capillaries make the content to be filtered out properly. There are actually 3 steps by which the excretion occurs, the urine formation occurs. So now let's deal with the structure as well as along with that the function of this particular nephron. So now here you can see the kidneys, the bean-shaped kidneys, they are present in the body, and this white color structure overhead is known as the adrenal gland. The gland which is very much functional in our body to secrete some of the hormones. Now this area is a cortex area. This area is a medullary area. The medullary area and the cortex, they have the structure known as nephron. So you can see the red color afferent arteriole, the yellow color Bowman capsule, the cup-like structure, the glomerulus being fitted inside this particular glomerular Bowman's capsule. Now this is the proximal convoluted tubule, then the descending limb of loop of Henle, the ascending limb of loop of Henle, then comes the DCT, the distal convoluted tubule, and finally leading its content into a long tube known as collecting duct. Now when you move back, when you see how does this process of the urine formation occur, you can see this is the nephron. This is having the 3 steps by which the urine formation occurs. The first step is the glomerular filtration. As the word says, glomerular. So major role that is being taken part is by the glomerulus. The second is the tubular reabsorption. Now the tubular means here the tubule, the PCT, the DCT, the proximal convoluted tubule, the distal convoluted tubule, they are the one which are going to take part in the process of filtration. Now, the third is the secretion thing. The secretion which will help the tubule to take out the contents from the blood, so that कुछ रह गया है तो वो भी filter out हो जाए, and the urine, the filter so form, the urine so form should be drained into the content of this particular tube. So you can see here the afferent arteriole bringing the blood which is full of the contents like the glucose, amino acids, the ions like sodium, potassium, bicarbonate, chloride ions, the urea, creatinine, etc. These all contents are present in the blood. The body needs to expel out the urea. Okay. So now when this afferent arteriole, it drains its content into, this is the afferent arteriole draining its content into this capillary, the Bowman's capsule or the glomerulus. Imagine करो you have a pipe, ठीक है, और उस pipe का nozzle आपने दबा दिया आगे से हल्का सा. What will happen? The pressure will be there. So when this is a large capillary, the large blood vessel, when it breaks down into the smaller capillaries with the pressure, they start filtering up the blood. And since the pore size, it decreases, so it will not allow the proteins to move inside. So the contents of this particular filtrate, they are same like that of a plasma. So we call it as a deproteinized plasma. The filtrate that is formed in the Bowman capsule is deproteinized plasma. Now this content of this deproteinized plasma, they move into the coil structure known as the distal convoluted tubule. This distal convoluted tubule, see, body needed that blood should expel out the urea. But what did the blood do? What did this afferent and glomerulus have done? They've given everything to the nephron. You do your work. Now what happens? This DCT now realizes that I have given everything. I need to absorb it back. I just want the urea to get expelled out, not everything. So there occurs the absorption, the reabsorption of the things. The reabsorbed things, now the content so form filtered now moves from the loop of Henle to the upper position, that is the DCT, where the secretion of the things occur. The secretion here, now if I talk of the urine so form is acidic in nature. The body does not want extra ions to be present in the body. So the body in the form of blood carried out by the arteries going to the nephron, they want this thing to expel out. And this expelling out of the things is done actually by this particular DCT. Now this secrete is blood में से tubule में डालना. Reabsorption is nephron, means tubule में से blood में डालना. Now the hydrogen ion, potassium ion, they are secreted into the DCT. And now when this filter will move into this collecting duct, then we'll be saying it as the urine. Till now the word filter was used, the filtrate was used. Now let's again see the mechanism. So here you can see this is the glomerulus, this is the afferent arteriole draining its content into this glomerulus, the tuft of the capillaries, and the lining of this particular afferent arteriole and the Bowman capsule, they are made by the epithelial tissue. The blood moving inside, then the things, they get filtered now. The blood is getting filtered, you can see. Here you can see the things, they are into the blood. Now from blood, they are moving into the cup-like structure. Now the things which are moving to this cup-like structure, they are having now the filtrate, glomerular filtrate. Once it was blood, till it was in the glomerulus. Once it reaches the Bowman capsule, it is known as filtrate. Now this filtrate has water, it has inorganic ions like hydrogen, potassium, bicarbonate, sodium, chloride, various ions that we take through the food, the minerals we take through the food, they are broken up into ions, and now if they are extra, they need to be moved out from the body. So now this glucose, amino acid, they are along with the metabolic waste, that is urea and the creatinine, they are expelled out. They are moved out. Now, after the filtration has occurred, the second step that occurs is the tubular reabsorption. So, dealing with this tubular reabsorption, you can see now this efferent arteriole, it has formed a network over the nephron. जाल बिछा दिया, ठीक है, ताकि जो भी चीज है जो एक्स्ट्रा दे दी थी, that should be moved out, that should be taken out from this particular thing. So taking out of this content, you can see here now the things that get reabsorbed now are the sodium, the water molecules, the chloride ions, the glucose, amino acid, and urea, and all this is occurring in the PCT. This is the PCT, the yellow color PCT, and the red color is again in the capillary. The capillaries which are carrying the oxygenated blood. Along with that, the process of circulation will also occur. The process of respiration will also occur. They'll give oxygen to the PCT because उसमें भी cells हैं. That is why this arteries they change into vein once they give the content to the oxygen to the nephron. So here you can see there is the absorption of sodium, water, chloride, glucose, amino acid, and urea. Some amount of urea again goes back to the body, and that is why the blood needs to be get filtered again and again. 1 minute में it's you can say हमारी body में 5 liter blood है. So in 1 minute 1 liter of the blood gets filtered. Or when it is getting filtered, you can say जब filtration हुई, the glomerular filtrate जब बना, वो बनता है in a whole day. If I talk of, it forms 180 liter per day of filtrate. अगर reabsorption ना होता तो blood is 5 liter. और पूरे कंटेंट को अगर हम देखें, the filtrate को अगर हम देखें, तो the filtrate formed is 180 liter per day. क्या होगा? 125 ml you can say per minute. सारा ही ब्लड खत चला जाएगा into the urine, and that is why the reabsorption occurs. यहाँ पे सारा content जो है वो कहाँ डाल दिया जाता है? Into this particular arteries, the capillaries. He reabsorb them. One thing is important, the amount of water that we drink. जितना ज्यादा वाटर इनटेक करोगे उतना यूरिन फ्लशिंग आउट ज्यादा होगा, उतना यूरिया जो है वो क्या होता जाएगा? जल्दी जल्दी एक्सपेल आउट होता जाएगा. Now this expelling up of this particular urea from the body is the main thing that is to be done in the urine formation. So the absorption, the reabsorption of this thing is occurring in the PCT and some amount in the DCT also, in the loop of Henle also. Amount of water will decide the amount of urine formed. The amount of water will determine the amount of urea so expelled out. Now you can see the third thing that is occurring is the tubular secretion, which occurs in the DCT, and it helps in maintaining the water balance of the body, the water ionic balance of the body. Now here from the blood, as I said, secretion means blood से tubule में आना. Reabsorption was nephron से blood में जाना. So if I am talking of this, you can see here the contents of this particular nephron, the blood, like hydrogen ion, some of the drugs, some of the waste material, jo still they were left, the potassium which were left, they are now poured down into this particular structure, that is the nephron, the DCT. Now this still filtrate. Now the H plus ion, the content increases. जितना ज़्यादा H plus होगा उतना acidic ज़्यादा होगा. So urine is acidic in nature. Now the next is Now concluding this, what is